Inside your arteries right now, billions of red blood cells are rushing past, and on the surface of every single one of them sits a microscopic identification system that dictates exactly who you can safely exchange blood with. Austrian immunologist Karl Landsteiner first mapped the visible effects of this system in 1901. He observed that mixing serum and red cells from different people caused them to clump together in predictable patterns, leading him to classify human blood into groups A, B, and O. Mapping these patterns requires looking past simple inheritance and examining how three distinct alleles at a single locus interact with an independent sequential biochemical pathway. This diagram displays the human karyotype, allowing us to trace the architecture to its precise genetic origin. If we zoom into the long arm of chromosome 9, right at position 9q34.2, we find the ABO gene. At this locus, the alleles do not code directly for antigens. Instead, they code for glycosyltransferases, specialized enzymes that function by modifying existing carbohydrate structures on the cell membrane. The macroscopic blood types we test for in the clinic are the visible downstream outcomes of these microscopic enzymes executing their specific genetic instructions. To observe how these enzymes build your blood type, we have to start at the biological baseline, the bare lipid bilayer and proteins making up the erythrocyte membrane. This 3D molecular model is our starting point. Before the ABO gene enters the picture, the FUT1 enzyme attaches L-fucose to a precursor disaccharide extending from the cell membrane. This newly formed carbohydrate structure is the H antigen, the obligate universal precursor required to build the rest of the ABO system. Once the H antigen is established, the ABO locus takes control. In the human population, two dominant genetic blueprints compete here, the IA allele, which codes for the GTA enzyme, and the IB allele, which codes for the GTB enzyme. These two transferase enzymes are nearly identical. Out of 354 amino acids, they differ by exactly 4 residues. That minute structural variation dictates their entire function. The GTA enzyme's specific geometry allows it to grab M-acetylgalactosamine and attach it to the H antigen backbone using an alpha-1,3 glycosidic linkage. Conversely, the GTB enzyme's slightly altered shape grabs D-galactose, attaching it to the exact same position on the H antigen using the identical alpha-1,3 linkage. A tiny genetic polymorphism alters the enzyme's substrate specificity, creating divergent biological antigens from the same starting material. This diagram maps the logical correlation between genetics and clinical blood types, organizing the 6 possible human genotypes alongside their resulting macroscopic phenotypes, surface antigens, and plasma antibodies. We begin with complete dominance. Both the IA and IB alleles are dominant over the recessive lowercase I allele. Inheriting even one functional IA or IB allele produces enough transferase to yield a straightforward type A or type B phenotype. If an individual inherits both an IA and an IB allele, both functional transferase enzymes are manufactured. Neither enzyme masks the expression of the other. Both actively modify the H antigens on the cell surface at the same time, producing a textbook biological proof of genetic codominance. There are strict structural gradients within these classifications. Type A blood is further divided into A1 and A2 subgroups. A1 cells pack roughly 1 million A antigen sites onto their surface, while A2 cells express only a quarter of that density, leaving a large amount of residual, unconverted H antigen intact. Finally, we encounter the homozygous recessive II genotype. This allele is amorphic. It produces no functional transferase whatsoever, leaving the universal H antigen unmodified on the cell surface, a condition we recognize clinically as type O. The completed diagram connects hidden genetic variation to visible phenotypic identity. While the antigens are anchored to the red cell membrane, the clinical consequences of blood grouping are determined by the isoagglutinins circulating in the plasma. This animation illustrates Landsteiner's law. The human body naturally generates isoagglutinins, specifically multivalent IgM antibodies, against whichever ABO antigens are missing from its own cell membranes. These antibodies form early in life. The immune system detects gut commensal bacteria carrying terminal sugars that share structural motifs with human A and B antigens. If your own cells lack those specific sugars, your body treats the bacterial motifs as foreign, arming your plasma with antibodies against them. Routine diagnostic slide tests leverage these exact IgM antibodies. Because an IgM antibody has 10 binding sites, introducing it to mismatched red cells forcefully cross-links the cells into a highly visible, insoluble lattice. In advanced research settings, we use specific plant proteins called lectins to detect these antigens. Locus bifloris binds the A antigen, Griffonia simplifolia isolates the B antigen, and Ulix europaeus marks the H antigen. These rules of immune recognition define transfusion medicine. Type AB plasma contains 0 antibodies, permitting those individuals to act as universal red cell recipients. Conversely, Type O red cells lack both A and B antigens, allowing them to bypass a recipient's antibodies as the universal donor. These rules are perfectly consistent until we analyze a paradoxical clinical scenario: a patient who genetically inherits fully functional IA or IB alleles, but consistently tests serologically as type O. This diagram displays a standard Mendelian cross between a heterozygous type A and a heterozygous type B parent. The model predicts a perfectly distributed 1-to-1-to-1-to-1 probability for A, B, AB, and O offspring. In 1952, Dr. Y.M. Bende and his colleagues documented a break in this predictive model in Mumbai, discovering an anomaly now known as the Bombay phenotype, or OH. This metabolic cascade chart reveals the epistatic block causing the anomaly. If an individual inherits a recessive mutation at the FUT1 locus, the HH genotype, their body cannot synthesize the foundational H antigen. This halts the sequential pathway before the ABO gene can interact with it. Biological pathways enforce strict hierarchies. Without the H antigen precursor existing on the membrane, the downstream genetic instructions for A or B enzyme production are rendered meaningless. At the molecular level, the failure is absolute. The fully functional GTA and GTB transferase enzymes are present, but with 0 H antigen substrate, they simply float above the bare lipid bilayer. Because this patient's cell membranes are devoid of A, B, and H antigens, their immune system fails to develop self-tolerance for these structures. This results in the production of anti-A, anti-B, and the unique anti-H antibody. This creates a life-threatening complication during transfusion cross-matching. A standard laboratory test will identify a Bombay patient as type O, but because of their anti-H antibody, giving them normal type O blood, which is heavily coated in H antigens, would trigger a fatal immune reaction. Bombay individuals can only safely receive blood from other HH donors. We can now update the master diagram. The Bombay phenotype is appended at the bottom, representing a case where an upstream mutation overrides the expected ABO inheritance. The ABO blood group demonstrates how a single polymorphic locus, when paired with an upstream biosynthetic step, determines the precise immunological identity of every human cell.